You shower every single morning. You change your shirt before you leave the house. You do laundry more often than you ever did in your 30s. You spend money on quality soap, quality deodorant, quality everything. And yet, there it is. That look. The one your grandchild gives you when they lean in for a hug. That almost invisible moment when your son or daughter-in-law takes a small step back during a conversation and pretends nothing happened. You see it. You notice. And it quietly chips away at something deep inside you. But here's the part that nobody talks about. At night, after you've undressed for bed, you catch it yourself. Not the smell of sweat, not the smell of something dirty. Something stranger than that. Something that reminds you of an old library book pulled off a shelf that hasn't been touched in decades. Or the dusty attic of a farmhouse. Or a bottle of cooking oil that was forgotten at the back of a cupboard for 2 years. You shower again the next morning and it's gone for an hour. Maybe 2. Then it comes back. And here is what nobody tells you, and what I am going to tell you today. No amount of showering will fix this. Not because you aren't clean, but because this smell is not coming from your skin's surface. It is being manufactured from somewhere much deeper, and there is a name for it, a molecular formula, a proven biological mechanism behind it, and most importantly, there are specific, science-backed ways to address it that most hygiene advice never mentions. Stay with me. Because what you're about to learn isn't just about smell. It's about what that smell is actually telling you about your body's internal chemistry, and how listening to it might be one of the most useful things you do for your health this year. To understand what's really happening, we need to travel back to the year 2001, a laboratory in Japan, a research team led by a scientist named Shinichiro Hayes. The question they wanted to answer sounded almost embarrassingly simple. Does the smell of old age actually exist as a physical, measurable phenomenon? Or is it just a social myth, a bias that people project onto elderly individuals? To find out, they recruited a group of volunteers ranging in age from 26 to 75. Each volunteer was given a specially prepared cotton t-shirt, treated to maximize absorption of everything the skin releases overnight. The participants wore these shirts to bed. The next morning, the shirts were collected and sent to an instrument called a gas chromatograph. If you've never heard of a gas chromatograph, here's a simple way to think about it. Imagine you could take a full orchestra and separate out every single instrument playing every single note simultaneously, and write down exactly which instrument played which note at which moment. That's essentially what a gas chromatograph does with smells. It takes a complex mixture of airborne molecules and separates it into its individual chemical components, identifying each one by name. When the team compared the chemical profiles of the young volunteers against those over 40, they found something they had not expected. There was a specific compound present in significant quantities on the skin of older participants that was virtually absent in younger ones. That compound has a name, 2-nonenal. Write it down if you want. But more importantly, remember the concept, because 2-nonenal is the central character in everything that follows. You don't need a chemistry lab to recognize 2-nonenal. You have already encountered it dozens of times in your life. You just didn't know what it was called. Think about the smell of an old paperback book that's been stored in a box in the basement. Think about the smell of cooking oil that has gone rancid. Think about the particular mustiness of a room that has been closed up for a long time. Not mildew, not mold, something more subtle and papery and waxy. That is 2-nonenal. What the Japanese scientists discovered is that this molecule is not imaginary, not culturally constructed, not a product of poor hygiene. It is a real measurable chemical compound with a precise molecular formula. It can be quantified in parts per million. It can be graphed over time. And crucially, it appears on the skin in a pattern that correlates tightly with age. People under 40 produce essentially none of it. After 40, it begins to appear. By the time a person reaches their 60s and 70s, it has become one of the dominant volatile compounds released from the skin's surface. But the most important finding, the one that changes everything about how we think about this, was not the existence of 2-nonenal itself. It was the answer to the next question: where does it come from? The scientific team had an initial hypothesis that turned out to be wrong, and understanding why it was wrong is the key to understanding the whole puzzle. Their first guess was that 2-nonenal was produced by bacteria on the skin. That would be the logical assumption because that's how most body odor works. Bacteria that live harmlessly on the skin break down components of sweat and produce smelly byproducts as part of their metabolic activity. If older skin smells different, maybe older people have different bacteria. So the team tested for this. And the results were surprising. Bacteria were not the primary cause. 2-nonenal was being generated directly on the surface of the skin through a purely chemical process. No microbes required. It is produced through oxidation, the same fundamental chemical reaction that turns a cut apple brown within minutes, the same reaction that makes iron rust, the same reaction that causes cooking oil to go rancid. Only here, it's happening on your skin. Now let's understand exactly how. Because once you understand the mechanism, the logic of the solution becomes completely clear. Your skin is constantly covered by a very thin film of oil secreted by your sebaceous glands. This sebum film is not a sign of uncleanliness. It is an essential protective barrier. It holds moisture in, keeps pathogens out, and maintains the structural integrity of the skin. Without it, skin would crack and lose its ability to heal. Here's where age enters the picture. The composition of this sebum film changes over a lifetime. In younger adults, it contains a certain mixture of fatty acids, but after the age of 40, the body begins producing higher concentrations of a particular class of compounds called omega-7 fatty acids, specifically palmitoleic acid and vaccinic acid. In younger people, these are present only in trace amounts. By the time someone reaches their 60s, these omega-7 acids have become a major component of the skin's oil layer. By themselves, these acids are odorless. They're simply raw material sitting on the skin's surface. But when they are exposed to oxygen, which being on the surface of the skin, they constantly are, they begin to oxidize. And when they oxidize, they break apart. One of the small, volatile fragments that breaks off is our molecule, 2-nonenal. 2-nonenal is lightweight. It evaporates easily from the skin into the surrounding air. It travels directly to the nose of anyone who comes close. And here is what makes it particularly potent. The human nose can detect it at extremely low concentrations, just a few molecules per cubic centimeter of air. The threshold of perception is incredibly low. So why doesn't washing help? Because 2-nonenal is fat-soluble, not water-soluble. Water and ordinary soap rinse away some of the oil on the skin surface, but the sebaceous glands immediately begin producing more oil. Within an hour or two, a fresh layer of fatty acids is on the skin's surface, and oxidation begins again. You can't wash away a continuous chemical process. You can only interrupt it briefly. This is why the smell comes back so reliably after a shower. It was never just sitting on the surface waiting to be washed off. It is being manufactured in real time, so we know the raw material. Omega-7 fatty acids accumulating in the skin's sebum layer as we age. And we know the trigger, oxidation. But why does oxidation happen so much more aggressively after 50? The answer lies in something called the antioxidant defense system. Think of it this way. Inside every square centimeter of your skin, there is a standing army of molecules whose sole job is to intercept and neutralize a class of unstable, highly reactive particles called free radicals. Free radicals are generated constantly by sunlight, by air pollution, by stress, by the natural byproducts of the body's own energy production. If left unchecked, they crash into the fatty acids in your skin's oil layer and trigger a chain reaction of oxidation, like knocking over the first domino in a very long row. In a young adult, this army is strong and fully staffed. Vitamin E, coenzyme Q10, glutathione, a full suite of enzymatic defenders, all working in rotating shifts, neutralizing free radicals before they can do significant damage. With age, this army shrinks. Vitamin E levels in the skin decline. The enzymes that produce antioxidant compounds become sluggish. The body's overall capacity to repair oxidative damage decreases. The defenders are outnumbered, and when two conditions exist simultaneously, the presence of omega-7 fatty acids as raw material and a weakened antioxidant defense system. The result is inevitable. Free radicals attack the omega-7 acids. The acids oxidize and fragment. 2-nonenal is released. This is not a disease. This is not a malfunction. It is a natural consequence of biological aging, predictable, measurable, and in significant part, addressable. Here is something that will genuinely surprise you. The research that followed Hayes's initial discovery revealed that human beings don't just develop one age smell. We actually pass through a sequence of distinct chemical olfactory signatures across our lifetimes, each one reflecting the specific biochemistry of that life stage. Research published in 2016 mapped out this progression in detail. Scientists collected skin secretion samples from volunteers across multiple age groups and analyzed the dominant volatile compounds at each stage. In people between roughly 30 and 40, the dominant odor compound is a molecule called diacetyl. If you want to know what diacetyl smells like, think of slightly sour butter that's been left too warm, a faintly sharp, creamy, fermented quality. This compound is produced not by the body's own chemistry, but by bacteria on the skin that feed on lactic acid in sweat. In active adults in their 30s, there is plenty of lactic acid and plenty of bacterial activity, and diacetyl is the result. It is, essentially, the chemical signature of the busy, stressed, hard-working middle-aged body. After 40, this production begins to slow. The bacteria that produce diacetyl become less active. Sweat composition changes. The lactic acid supply dwindles. The diacetyl era fades. And into its place steps 2-nonenal, produced through a completely different mechanism, arriving from a completely different biological process, announcing a new chapter. So the smell of age that people associate with elderly relatives is actually the third act in a sequence. Youth has its own smell. The working years have their own smell. The later decades have theirs. Each one is a chemical biography written on the skin's surface. In 2012, a research team led by investigator Suzanne Mitro conducted a study that produced one of the most counterintuitive results in the science of olfaction. The design was careful and rigorous. 3 groups of volunteers, young adults aged 20-30, middle-aged adults 45-55, and elderly adults aged 75-95, each wore specially prepared underarm pads for 5 consecutive nights. The pads were then sealed in glass jars and presented to a separate group of evaluators who knew nothing about the ages or identities of the donors. This was a true blind test. The evaluators were asked 2 things. First, could they identify the age group of each sample? And second, how would they rate the pleasantness or unpleasantness of each smell? The first result was expected. Yes, people could detect age-related differences in smell with reasonable accuracy. The smell of age is real and perceptible. The second result was not expected at all. When evaluators rated the pleasantness of each group's odor, the elderly group, particularly elderly men over 75, was rated as producing the least unpleasant smell of all 3 groups, not the worst, the best. The odor associated with young adult men was rated as significantly more intense and more unpleasant than that of elderly men. The sharp, musky, sometimes aggressive quality of young male body odor ranked lower on the pleasantness scale than the softer, more subdued scent of older bodies. The researchers drew an important conclusion from this. The cultural stigma attached to the smell of old age has no solid biological basis. In a neutral laboratory setting, stripped of all social context and preconception, the smell associated with aging is not repulsive. It is simply different, quieter, less sharp, more subtle. The negative feelings that many people associate with this smell are learned, not innate. They have been shaped by decades of advertising that equates any body odor with failure, by a culture that has systematically pushed the reality of aging to the margins. Understanding this matters enormously for the psychological weight it can lift from you. You are not failing at hygiene. Your body is not betraying you. It is moving through a natural biological transition with its own chemical signature, one that is actually, in objective laboratory conditions, milder and less offensive than the odor of a 25-year-old after a long day. Everything we have discussed so far would be scientifically interesting, but practically limited. if 2-nonenal were simply a harmless odor molecule, if it just floated off the skin into the air, and that was the end of it. But research from 2021 through 2024 revealed something much more significant. 2-nonenal is a member of a chemical family called aldehydes. Aldehydes are highly reactive. They don't just float passively, they form chemical bonds with other molecules, particularly with proteins. Studies published in biochemistry journals demonstrated that a portion of the 2-nonenal produced at the skin's surface doesn't evaporate at all. It stays, and it attacks the skin cells themselves, specifically the cells called keratinocytes, which are the primary structural building blocks of the skin's outer layer. When 2-nonenal binds to these cells, it triggers a process called apoptosis, programmed cell death. The cell effectively receives a chemical signal telling it to shut down. This happens slowly, invisibly, day by day and year by year. But the cumulative effect is measurable. Skin that becomes progressively thinner, drier, slower to heal after minor injuries, less elastic. In other words, the very compound that signals skin aging is also actively accelerating that aging. It is not merely a symptom, it is in part a cause. And the story goes deeper still. 2-nonenal is also capable of entering the bloodstream. Once there, it can bind to one of the body's most important transport proteins, albumin. Albumin is sometimes described as the body's freight system. It carries hormones, nutrients, and medications through the blood and removes metabolic waste products. When 2-nonenal binds to albumin, it forms what biochemists call an adduct, essentially a damaged version of the protein. The immune system, which monitors blood proteins for signs of damage, identifies these modified albumin molecules as suspicious. It tags them for removal and ramps up its surveillance activity. Over time, this contributes to a state of chronic, low-grade systemic inflammation. This is the iceberg beneath the surface. The smell of 2-nonenal is the visible tip. Beneath it lies a much larger structure, the process that doctors call oxidative stress, the slow, progressive imbalance between free radical production and the body's ability to neutralize it. Oxidative stress is not a minor inconvenience. Medical research has linked it to atherosclerosis, the buildup of plaque inside artery walls, to the clouding of the eye's lens that causes cataracts, to the kind of cognitive decline that gradually impairs memory and processing speed, and to the chronic joint inflammation that makes climbing stairs an exercise in willpower. So when your body begins producing more 2-nonenal, when that particular smell becomes noticeable to you or to those around you, your body is actually doing something remarkable. It is sending you a signal, a very specific, very measurable signal that says, the antioxidant balance inside this system has shifted. The protective forces are being outpaced. Most people dismiss this signal as a cosmetic embarrassment. Wise people will recognize it for what it is: diagnostic information, a warning light on the dashboard that is worth paying attention to. When Japanese researchers understood that conventional soap was ineffective against 2-nonenal, because the molecule is fat-soluble, produced continuously, and not caused by bacteria, they began looking for substances that could neutralize it chemically, on contact. The solution that emerged from their research came from a fruit that has been central to Japanese culture for over 1,000 years. The Japanese persimmon, Diospyros kaki, the ripe orange fruit that appears in markets in late autumn. The key compounds in persimmon, particularly in its skin and unripe flesh, are called tannins. Tannins are the molecules responsible for that characteristic mouth-puckering, tongue-coating astringency you experience when you bite into an unripe persimmon, drink strong black tea, or sip a bold red wine. The sensation is not imaginary. Tannins are physically binding to the proteins on the surface of your tongue, and this binding ability is precisely what makes them effective against 2-nonenal. Tannins have a demonstrated capacity to bond chemically with aldehyde molecules, and 2-nonenal is an aldehyde. When persimmon extract is incorporated into soap or body wash, the tannins it contains don't just rinse the skin. They react with 2-nonenal on contact, converting it from a volatile, smell-producing compound into a stable, non-volatile, odorless compound that washes away cleanly. Laboratory testing showed this approach neutralizes up to 97% of 2-nonenal present on the skin's surface, not masking it with fragrance, not washing it off with water, chemically disarming it. This technology has been standard in Japanese personal care products for older adults for years. In the West, it is only recently gaining recognition. A parallel line of research, published in 2024 in a Journal of Molecular Biochemistry, investigated compounds found in a much more ordinary vegetable, the eggplant, aubergine. Scientists identified specific phenolic compounds in eggplant called phenolamides, in particular a molecule called N-trans-ferroyl putrescine. These phenolamides were found to do two things simultaneously. They bind directly to 2-nonenal and neutralize it, similar to persimmon tannins, and they also reduce the concentration of free radicals inside skin cells, meaning they address both the smell and one of its root causes at the same time. Both of these approaches work from the outside in, but to address the problem at its source, you need to work from the inside out. The oxidative process that produces 2-nonenal begins inside the body, driven by free radicals and a depleted antioxidant system. Fortunately, the body's antioxidant defenses are significantly influenced by what you eat. This is not vague wellness advice. It is specific, measurable, biochemically documented. Direction 1. Anthocyanins. Anthocyanins are the pigments that give fruits and vegetables their deep blue, purple, and dark red colors. They are found in blueberries, blackberries, red cabbage, beetroot, and dark grapes. But the absolute richest known dietary source of anthocyanins is black currant. Japanese researchers ran a clinical trial in which middle-aged and older adults consumed black currant powder daily for several weeks. At the end of the trial period, the concentration of 2-nonenal being released from their skin was measured and compared to baseline. The result was statistically significant. 2-nonenal emissions had meaningfully decreased. What this tells us is something profound. Antioxidants consumed in food travel through the digestive system, enter the bloodstream, and reach the skin, where they actively suppress the oxidative chain reaction that produces 2-nonenal. The skin is not a sealed container. It is a living organ in constant chemical communication with everything you put into your body. Direction 2. Catechins from green tea. Catechins are a specific class of antioxidants found in concentrated form in green tea, particularly in unfermented, lightly processed varieties. The fermentation process used to make black tea destroys most of these catechins, which is why green tea has markedly higher concentrations. Catechins have been shown in controlled studies to directly inhibit lipid peroxidation, which is the technical term for the oxidation of fatty acids that produces 2-nonenal. 2 to 3 cups of quality green tea per day provides a clinically meaningful antioxidant benefit. One crucial detail: brew green tea at temperatures no higher than 80 degrees Celsius, about 175 degrees Celsius. Water that is too hot degrades the catechins before you even drink them. Let boiling water sit for 3 to 4 minutes before pouring it over the tea leaves. Direction 3. Vitamin E. Vitamin E is the body's primary fat-soluble antioxidant, which means it operates specifically within fatty environments, including the oil layer on the skin's surface. It is positioned exactly where it is most needed, at the interface where free radicals meet fatty acids. The best dietary sources of vitamin E are cold-pressed, unrefined vegetable oils, especially wheat germ oil and sunflower oil. Nuts, almonds and hazelnuts are especially rich. Sunflower seeds, avocado, and leafy green vegetables. A diet consistently low in these foods leaves the skin's antioxidant defense substantially undermanned. Direction 4. Omega-3 fatty acids. Omega-3 fatty acids, found in fatty fish, salmon, sardines, mackerel, flax seeds, and walnuts, perform two important functions in this context. They reduce systemic inflammation, the chronic, low-grade inflammatory state that is aggravated by oxidative stress, and they help stabilize cell membranes, making them more resistant to oxidative damage. Direction 5. What to reduce. Every intervention aimed at strengthening antioxidant defenses will be partially undermined if the sources of free radical load are left unaddressed. Smoking is the single largest discretionary contributor to free radical burden. A single cigarette triggers a massive, sustained surge of oxidative stress that takes the body hours to recover from. If you smoke, no nutritional strategy will be fully effective until this is addressed. Excess sugar and refined carbohydrates promote a process called glycation, a different form of protein damage that compounds the effects of oxidative stress. Chronic sleep deprivation interrupts the body's nighttime repair cycle, during which the antioxidant systems replenish themselves. Most of the enzymatic repair work happens between 11 p.m. and 3 a.m. during deep sleep stages. Consistently shortened or fragmented sleep means this work doesn't get done, and excessive sun exposure without protection floods the skin with UV-generated free radicals, exactly the force that triggers the oxidative chain reaction we are trying to prevent. There is one more dimension to this that rarely gets mentioned, and it makes a considerable practical difference. 2. Nonenal, being fat-soluble, binds readily to synthetic textile fibers. Polyester, acrylic, nylon. These materials are essentially molecular sponges for fat-soluble compounds. Once 2-nonenal is absorbed into synthetic fabric, it bonds with the fiber in a way that ordinary washing does not fully break. You launder the garment, hang it in the wardrobe, wear it a week later, and the smell returns within hours of putting it on. Not because you are generating it faster, but because the garment never fully released what it had captured. Natural fibers behave differently. Cotton, linen, silk, and wool do not hold fat-soluble compounds in the same way. They breathe, they release volatile molecules during washing, and they do not accumulate a long-term odor reservoir in their fibers. Transitioning your everyday wardrobe, particularly the clothes worn closest to the skin, toward natural materials is not an aesthetic preference. It is part of a practical odor management strategy. Similarly, 100% cotton bedding, changed more frequently than you might currently be doing, removes the residue that accumulates overnight when the skin is in close, sustained contact with fabric for 7 or 8 hours. When all the research is assembled into a coherent picture, the approach has a clear architecture. From the outside, you use cleansing products that contain persimmon tannins or eggplant phenolamides, compounds that chemically neutralize 2-nonenal on contact rather than simply washing the skin's surface. These are not novelty products. They are based on well-documented biochemistry and have been available in Japan for many years. From the inside, you systematically strengthen the antioxidant defense system through diet. Dark berries and black currant for anthocyanins, properly brewed green tea for catechins, nuts and cold-pressed oils for vitamin E, fatty fish or flaxseed for omega-3, and green leafy vegetables for their broad antioxidant content. You reduce the factors that fuel oxidative stress: smoking, excess refined sugar, chronic sleep debt, and unprotected prolonged sun exposure. You transition your clothing to natural fibers: cotton, linen, silk, wool, particularly for garments worn against the skin and for bedding. And you approach all of this not as a crisis response or a cosmetic fix, but as a sustainable, ongoing practice of physiological self-care. None of these changes requires a dramatic life overhaul. They can be introduced gradually, 1 or 2 at a time. The body's response to sustained, consistent nutritional and lifestyle change is slow, but reliable. Within 4 to 6 weeks of implementing even a few of these steps, most people begin to notice a shift in the smell of their clothing at the end of the day, in the freshness of their sheets in the morning, sometimes in a subtle brightening of overall energy and mental clarity. Here is the thought I want to leave you with, because it is the most important one. The molecule 2-nonenal is not your enemy. It is a messenger. It is the body's chemical way of communicating a state of internal imbalance. Specifically, an imbalance between the forces of oxidation and the forces of antioxidant protection. The same oxidative stress that expresses itself as a change in body odor after 55 is also at work, more silently, less detectably, in the walls of blood vessels, in the neurons of the brain, in the tissues of joints and eyes. Oxidative stress is not a local skin problem. It is a systemic biological state that affects every organ simultaneously. When you act to reduce it, through diet, through lifestyle, through targeted topical care, you are not just managing a social inconvenience. You are actively participating in the maintenance of your cardiovascular health, your cognitive function, your cellular repair capacity, and the integrity of your immune system. The smell was just the signal you could detect. What you are actually addressing is far larger. The Japanese scientists who began this work in 2001 were trying to answer what seemed like a narrow question about odor. What they ultimately uncovered was a window into the biochemistry of human aging itself. A window through which, if you look carefully, you can see not just a smell, but a whole system, and not just a problem, but a set of very practical, very accessible tools to work with. You shower because you understand the connection between cleanliness and health. You sleep because you understand the connection between rest and recovery. Now you have a new understanding. The foods you choose, the tea you drink, the oils you use, the fabric against your skin. These are not trivial daily choices. They are the inputs that determine the chemical environment inside your body, day by day, year by year. The body responds to what it is given, at any age, without judgment about how many years have passed. It simply reads the inputs and responds accordingly. Give it the right inputs, consistently, without urgency, without drama, and it will tell you in its own quiet way that it received them. The smell will soften. The skin will strengthen. The mornings will feel a little lighter. The years ahead will look a little clearer. That is what the science says is possible, and that is worth knowing.